We'll continue to Robyn Ball, who is a computational scientist at the Jackson Laboratory, present GenomeMUSter, which you'll explain. Yes. So thank you very much. Take it away.
Thank you. Can everyone hear me okay? I'll try to speak up. So my name is Robyn Ball. I work at the Jackson Laboratory. On the screen, you can see that I'm going to talk about GenomeMUSter. I'm really excited about it. It's a new comprehensive mouse variation database that you can use for your analyses. And the people that work closest with me on this is Hongping Liang. He's a software engineer at the lab. Molly Bogue, who many of you probably know, the mouse phenome database, Vivek Phillip, and Alyssa Chesler. She speaks for herself, I would say. Let's see here. Okay, but none of this was done in a vacuum. I think that's really important to say. There was a lot of time, a lot of effort, and a lot of expertise that went into this resource. In addition, we also had some variant data sets that were provided in the process. The BXD data set that Rob and Lou and David has worked on is integrated into Muster and is also available on the website. The CC data, the B6 data, and then Gary Peltz's lab gave us some long read sequencing data on lots of the inbred strains. So that is really what allowed us to do this work. And without our funding sources, we wouldn't be able to do any of it, right? So most of my effort was covered by the Cube Initiative at the Jackson Laboratory, but then a lot of the other collaborators across NIDA and the National Cancer Institute, and of course, here at the University of Tennessee. So the idea here is that when our software systems are connected, we can do great work, right? It becomes a knowledge graph of both analytic resources and data services. And that allows us to ask all the questions that we've been talking about in this conference, right? We can ask things like, what variants influence complex disease? And then if we find a gene of interest, is this a drug interacting gene? Is this a druggable target? What variants and genes have shared roles across species? So there's lots of questions that we can answer once we have these systems connected. And this is meant to be just a cartoon, not completely representative, but just to give you the idea. So what I'm going to talk to you about today is two components of this knowledge graph, GenomeMUSter and the meta-analysis service at the mouse phenome database. And this really allows us to answer the question of what variants in the mouse influence complex disease. So in order to do that, we need at least two things, right? We need a saturated grid of genomic information, and that's GenomeMUSter. And we also need data integration across multiple traits, and that's the meta-analysis service. So briefly, I'm going to go through this. GenomeMUSter was really built to harmonize data across many of the data sets that have been collected over the past few decades. There's a lot of genotype data as well as sequencing data. But of course, the genotype data especially is very sparse. It's difficult to analyze these data. If you've ever tried to do this, right, you have to exclude strains, exclude different SNPs, and try to get the data set merged in and harmonized so that you can actually do the work. So we're trying to do that for you so that you can use it as an analytical resource. So once we merge all these data together, there's a ton of gaps, right? And we need to be able to fill in those gaps with genotype imputation. So briefly, the way that we did this was we went within each 10 MB region. We calculated the phylogenetic distance across these strains. And we chose a set of predictor strains that was phylogenetically similar to the target strain, but also didn't have the same missing data, right? So it was a little bit of munging there. And then we used the Viterbi algorithm through HaploQA to do the imputations. And then we get this saturated grid, which is GenomeMUSter. And you can access it at muster.jax.org. So we have allelic states for 657 strains across 106.8 million sites. So this is really to show you the situation once we merge the data together. And so what you're seeing here, each of these rings in the circle is a data set that was incorporated into GenomeMUSter. And this is just talking about the merged set, not the imputed set. And So what's very evident here is that there are some data sets that have more dense data, but a lot of data sets that have very sparse data, as I was mentioning before. And so even like the largest data set that we have, which is the Sanger 2004 release, had about 80 million sites. That's really only 75% of the merge set, and it's only less than 10% of the strains that's in GenomeMUSter. And so this kind of gives you the sense of like how big the problem is in terms of really imputing and filling in the gaps. And so we really need an accurate imputation algorithm. We can fill in the gaps with noise, but we want something that we can actually use. So it actually worked quite well. We received a median accuracy of 0.944, and this is across all the strains. The way we did this was in each 10 MB region and for each strain, we had a held out test set that was not used in the imputation process. It was later merged in, but that gave us a way of evaluating how accurate we were in that local region, right? And then we can aggregate that. What I've done here is just aggregated those over the genome so you can get a sense of how accurate it is. So of course, the wild-derived strains are always have been difficult. There were some of them that we didn't do as well on, but as you can see in the purple there, some of them we actually did quite well. This will only get better as we add more data to GenomeMUSter. So after we've imputed the SNPs and filled in these gaps, we had a median of 91.6% of the strains were complete, which means that we had about 98 million SNPs for at least half of the strains. The minimum amount of sites that were covered was 85 million, which is 10 million bigger than Sanger, and it's across all 657 strains. So we have a very nice, dense, saturated GWID that we can use in our analyses. So you can access the resource either on the UI at muster.jax.org or via the API. So as well as GenomeMUSter, which is this merged data set, you can also access all the individual data sets that I mentioned. There's also available Sanger's Build 39 data set on the website. So however you want to use these data. The functional annotations were provided by the mouse variation resource at JAX, mvar.jax.org. And you can filter these data, right? So if you look in the bottom panel here, you can see that there's some darker blue and then some lighter blue color squares. So you can put that slider as far as the confidence threshold to where you're comfortable, right? So if you want to say, I only want SNPs with greater than 80% accuracy, you can set that confidence threshold, use the API or download the table, however you want to do it. This also is nice because it allows you to use local confidence information in downstream analyses. So if you wanted to weight your analyses, for example, you could do it that way. Okay, so now we've got this great SNP grid we can use in our meta-analysis. We also have recently integrated the meta-analysis service into the mouse phenome database. So it's integrated with these expertly curated mouse data, and that allows us to do a lot of really fun stuff, right? So because these data are expertly curated, we know the mouse strains, of course, we know the procedure that was used, if they were given a special diet, if they were given a special treatment, and we can integrate across all of these different populations and really get a lot of power in the meta-analysis to identify variants that are impacting complex traits and complex disease. And so what I'm showing you here on the screen, so these ontology terms, the parent term is response to addictive substance. So this is a meta-analysis across 397 measures. But we've also done individual meta-analyses on each of the substances. So this one is a response to cocaine, response to alcohol, response to nicotine, and a withdrawal response. And what you see here, we call the Fuji plot, right? And so each of these rings represents a different meta-analysis. So this blue ring is the response to addictive substance. The green is cocaine. The pinkish one is alcohol, and so on, right? We've got nicotine and withdrawal. And this is nice because it gives us a really bird's eye view of what's going on, not just within a single meta-analysis, but across all of these different substances. And I'll show you a little bit more here. So one of the things we did in the paper was we really wanted to show how muster can be used to characterize human GWAS using the mouse meta-analyses, right? And so one of our collaborators, Alex Hatum, just recently published this paper where he looked at multi-ancestry, multi-substance GWAS meta-analysis in humans. He identified 42 significantly associated genes. And so we said, hey, we've got that same kind of data in the mouse. Let's see where we see that shared signal. So of these 42 significantly associated genes that he found in his study, we are highlighting the ones where we also found them in the mouse, right? So what we can say is these multi-substance meta-analysis reveal the genes that have conserved roles across these species, right? So that's just the starting point. We find this in the human, that's great, but what does that tell us? Well, what we can do is we can use the mouse then, we find where there's conserved roles, and then we can go back to the variants in the mouse and really look at where those variants are affecting the traits. Which traits are they affecting? And so that's what I'm showing you here. So these are the human genes here on the left, and each of these lines is one or more variants that map back to traits, right? So what we can see here, if we look at this gene, PDE4B, which is of particular interest, we can see that it's related to withdrawal response, and it's across multiple substances, both alcohol and nicotine. And what we learned from this is when we really dug into the data, we learned that the effect increases as the dose of the drug increases, right? So what that tells us that we didn't know about the findings in the human is that we have a multi-substance withdrawal-mediated response with PDE4B, right? So that really allows us to get much more specific than we can do in humans. So I'm about out of time, so I just wanted to show you where you can get this stuff. So if you go to muster.jax.org, you can get to GenomeMUSter as well as all the 16 data sets that were harmonized in it. All of the references, all the information is there. Please cite our data contributors. You can also access Sanger and Build 39. If you go to phenome.jax.org, you can access the meta-analysis service that's integrated with these expertly curated mouse data. You can use the Fuji plot. And as I showed you those genes, right? Those genes that come from the variants are through the variant graph, Gene-Weaver's variant graph. And so the Fuji plot on MPD is integrated with that. So you can get the genes from the variants as well. So please contact phenome.jax.org with questions or contribute data. As everyone's been saying, we need more data, right? The data are what power these discoveries. You can read more about GenomeMUSter. It's on bioRxiv. We're in the review process. Please cite our data resources, I'll say that. And you can read more about the new tools and things that we have available at MPD through Bogue and Ball et al. And NAR, or a more recent one, mammalian genome, variant graph, etc. So thank you. Please grab me. I'm going to be around until this afternoon, or you can email me.